Hello, everyone. So I work with cutaneous T-cell lymphoma. So cutaneous T-cell lymphoma is a kind of disease where malignant T cells infiltrate the skin and creates this malignant phenotype of the skin. Cutaneous T-cell lymphoma is divided into two subclasses. One is skin-resident subclass, which is mycosis fungoides, and one is leukemic variant of cutaneous T-cell lymphoma, or LCTCL, which is Sézary syndrome. So I mostly worked with Sézary syndrome. Now, deregulation of STAT signaling pathway has been found in pathogenesis of variety of cancers, including cutaneous T-cell lymphoma also. So what we did low-pass whole genome sequencing. So low-pass whole genome sequencing of clonal PBMCs from these Sézary patients, of LCTCL patients, revealed copy number alterations in chromosome 17 in the STAT5 genomic region, which results in aberrant activation of STAT5. Now, since STAT5 is one of the critical mediators of different malignant properties of LCTCL, or Sézary syndrome, direct targeting of STAT5 holds significant clinical value. Here, we describe the preclinical validation of highly selective and potent agents. One is IQDMA, and another is derivatives of AC4-130 in comparison to the old or classical JAK inhibitors like tofacitinib and ruxolitinib. Now, this drug, AC4-130, was published last year in 2018 in a leukemia paper. It was synthesized by University of Toronto. And then we work with them, and they derive some more potent molecules from this AC4-130 to target SH2 binding domain of STAT5. Now, our workflow is pretty simple. The materials and methods of this workflow is quite simple. So what we do here is the Sézary patients came to our clinic. We take the blood from them. Then we isolate the malignant clones from the blood. We isolate the DNA from there, and it goes to whole exome sequencing. Now, this project have two parts. One part is to establish the correlation between copy number aberrations in chromosome 17 and STAT5 overexpression, and another part is to find out the targetable vulnerability of STAT5 in these patients. So let's talk about the first part. In the first part, the workflow is very simple. We take the blood, we isolate the clonal PBMCs, we isolate the malignant clones. These malignant clones, from there, we isolate the DNA and goes to whole exome sequencing. We also do qPCR and Western blots to confirm the data from whole exome sequencing. And the other part, which is here shown in next figure, what we do the drug discovery part, where we test the malignant clonal PBMCs with different drugs, or small molecule inhibitors, and check by cytotoxicity assays, qPCR, and immunoblot analysis how they are working. Now coming to the results. So this is figure 3. Now, this is the low pass whole exome sequence, whole genome sequencing data, where if you see in the chromosome 17 region particularly, so this chromosome 17 region, you can see that always this chromosome 17 region is highly gain. There is a gain. So out of our 6 patients, the chromosome 17 region is gained or duplicated most of the time. So in the patient 1, you can see there is a 26 MB gain. So this patient, in the P arm of a portion of the P arm of the chromosome 17 and the whole Q arm of the chromosome 17 is duplicated. Now, in the rest of the patient 9, 12, 21, all of these patients, we can see that the whole chromosome 17 Q arm is duplicated. And our last 2 patients, patient 17 and patient 10, they don't have actually chromosome Q arm duplication, but some downregulation in tumor suppression region. So in conclusion, out of 6, 4 patients have either huge gain or duplication in the chromosomal 17 region. And this exactly correlates with the data from the qPCR. So if you look here in the STAT5A and STAT5B region, we can see that STAT5A and STAT5B is highly overexpressed compared to healthy controls, but not STAT3 or STAT1. So we can directly conclude from there that the chromosome 17 STAT5 region is highly gained, duplicated, or overexpressed, and that's why the gene is overexpressed in these Sézary patients. Now, this is microRNA 155. So a recent paper a couple of years ago, it shows that STAT5 directly goes and binds to the microRNA 155 promoter region, which is B-cell integration cluster promoter region, and overexpress microRNA 155. And because STAT5 goes and binds to 155 promoter and overexpress, we use it as biomarker to check if the 155 is also upregulated in our patients. And what we can see is that yes, the microRNA 155, which is a designated oncomere, is upregulated in our patients compared to healthy controls. Now coming to the second part of the project, which is the drug discovery part, where we try to find out small molecule inhibitors which can inhibit the cutaneous T-cell lymphoma, particularly Sézary syndrome. So we took 2 Sézary syndrome cell lines, which is CX and HUT78, and 2 mycosis fungoides cell line, which is Myela and HNH. And we tested the drug. So pimozite is a classical STAT5 inhibitor, so-called STAT5 inhibitor, and we can see that pimozite have a very high IC50, 17, 21, 27. So it's not it's not a drug which we really can use as a clinic. Now AC4130, which is the classic, which was discovered last year, and it was potent for some other cell lines, but in the cutaneous T-cell lymphoma, it still have quite a high IC50. Now when we come to compound X, which is a derivative of AC4130, we can see it's have a pretty low IC50, 2 micromolar to 1 micromolar. Same for IQDMA. This is also have a IC50 for ranging from 2 micromolar to 900 nanomolar. And another derivative of compound AC4130, this is also have a pretty low IC50. Now when we check the Western blot, so this is one of the representative of Western blot of this drug testing, and it shows that the compound X doesn't change the total STAT5, but it particularly changes the phosphostat5. So with 5 micromolar of phosphostat, of this compound, the phosphostat5 is completely gone, where the STAT5 remains the same. So now coming to the last figure, where I try to compare. So I took one patient with 26 MB gain of chromosome 17 STAT5 region, and you can see that after the drug treatment with IQDMA, the IC50 is 3 micromolar. Now the patient 12, which is a chromosome 17 duplication, and the IC50 is 1 micromolar. But patient 10, which doesn't have a chromosome 17 STAT5 region gain or duplication, have an IC50 of 33 micromolar. And at the last, this is a healthy volunteer whose IC50 is 50 micromolar. So I want to highlight the therapeutic window of using these drugs. So here you can see a very nice therapeutic window. So this is inhibition of viability. And in all of the cases, you can see a very nice therapeutic window where there is a chromosome 17 STAT5 genomic region gain. But there is almost no therapeutic window where there is no chromosome 17 STAT5 genomic region gain. So from this, I'd like to conclude that there is a direct correlation between chromosome 17 copy number gain, STAT5 overexpression, and the targetable vulnerability of STAT5 by the selective inhibitors. And these highly selective derivatives of AC4130 and IQDMA emerges as a good candidate for targeting STAT5. So thank you for listening to my poster. Thank you.